So welcome. I'm really not here alone. Actually, I have lots of support. So I guess this is going to finish up soon, huh? So I'm here to talk to you guys about a research project that we just started this past fall here at TMCC. And I'm one of the PIs and instructors, and the other is Tina Sloan-Pomeroy, who's here in the front row. You'll meet her in a little bit. And we have about 15 researchers working with us on this project now, and several of them are here. Brianna Nicholson's here, and Jessica and Kathleen, and Jocelyn and Shayla, and they're going to show you some of their work towards the end of the presentation, so you can see kind of some of the stuff that we're going to be doing. TMCC has become part of the Phage Hunters program. It's a nationwide program. Students all over the country are searching for phage. How many of you heard of phage? So a couple people, mostly from our project and people who know us, all right? So do we need to turn a light on? Can you guys see the screen? It'll be, no. Is that too dark? Perfect? Okay. So phage are viruses. They're viruses that infect bacteria. And they're ubiquitous. They're found all over. Okay. And Phages is a program from the Howard Hughes Medical Institute. They paired up with the University of Pittsburgh. That's probably, in this country anyways, the biggest phage research group is from the University of Pittsburgh. And there's so many phages. I have some slides. We'll kind of introduce that in a minute. But Dr. Graham Hatfill, the PI working on the phages, said, there's no way in my lifetime I'll ever make a dent. So he thought, how can I have the biggest lab in the world? All right. And so he paired up with the Howard Hughes Medical Institute to start this program where he would recruit students from all over the country and get them all working on hunting phage, purifying phage, characterizing them, banking them, okay? And so he's probably got the biggest lab in the world. There's just close to 100 different campuses now join in the program, and more joining every single year. So we joined then the program this fall. It's our first semester teaching it. In July, Tina and I and Dr. Ellsworth went back for a week-long training on phage because, like you, we didn't know a lot about phage before. Okay? And so part of what we had to do is go out and collect soil samples from around, we were at the University of Maryland, and we had to go around and collect soil samples and bring them back to the lab and see if we could get phage. And so it was an intensive week, because we did all what we've been working on in a week. And so we actually were able to get enough phage growing, and I'll kind of show you a little bit how we do it, to put some on a grid and take an electron micrograph of it. And so this is actually the phage we found. It was from soil at Taxi Stop 4. The University of Baltimore, or University of Maryland, is like this big circle. We walked it, huh? It's a couple miles around, and there's taxi stops all around. So literally, we picked this up and we named it Taxi Stop 4. We haven't done any more work with this. We threw this away because we didn't have time to really do what needs to be done. But it was like the most exciting thing. It's almost like having a child. Swear, you go, you have your little teeny piece of little metal paper with maybe your phage on it, and you put it in the electron microscope, and you're just like, I hope something's there. It's like, no, wait till you guys do it. It's like you can't believe that there's really some kind of real particle there. Anyway, so that's kind of our history. So phage, what? Okay. Bacteriophages, like I said, are viruses that infect bacteria. There's 10 to the 31 phage particles on the planet. How many is that? It's a lot. It's like a trillion, trillion, trillion. There's some mathematicians in my clothes, right? It's a lot. And it's figured that there's like 10 to the 25 infections every second on Earth. So there's a lot of bacteria. And there's like 10 to the 30 bacteria on the planet. So there's more phage even than bacteria. So they're definitely everywhere. Do they really look like this kind of funky space capsule? Yeah, they amazingly really do. Okay? Viruses are really simple. They're just a piece of nucleic acid, DNA or RNA, and they're contained within a protein coat. The viruses that infect us aren't this spacey looking, okay? They tend to really just kind of be the head. They don't have the fancy tail, and right? So they're more just a little circular and with nucleic acid inside. So they're a little bit of nucleic acid, a little bit of protein, but they can sure do a heck of a lot of damage, right? And they do damage to us. We've all probably seen Ebola stuff recently. They put the hurt on bacteria too. Right? So the bacteriophages have the head. That's where the DNA or RNA is. It's a protein coat making that little capsid. They have this tail part. There's the shaft, and you can see seriously little landing legs, okay? Little spikes. And I'll show a brief animation kind of about how they work.
The T4 phage initiates infection of an E. coli bacterium by recognizing cell surface receptors of the host with its long tail fibers. A recognition signal is sent through the long tail fibers to the base plate. This unravels the short tail fibers that bind irreversibly to the E. coli cell surface. The base plate changes conformation and the tail sheath contracts, causing GP5 at the end of the tail tube to puncture the outer membrane of the cell. The lysosome domain of GP5 is activated and degrades the periplasmic peptidoglycan layer. The remaining part of the membrane is degraded, and DNA from the head of the phage can travel through the tail tube and enter the E. coli. After the life cycle is complete, the host cell bursts open and ejects the newly built viruses into the environment, destroying the host cell.
So just to give you kind of a little idea of how phages work, we'll look at some other pictures again later. Okay? So electron micrographs of phage in action. They really do kind of look like that. You can see the the cell wall of the bacteria with a little phage kind of getting themselves embedded in there. Okay? A whole bunch will attack one bacterium. Okay? And they'll just be loaded up on the surface. Okay? Inside, what viruses do, of course, all they've injected is a little piece of nucleic acid, right? Once inside the host cell, they take over the machinery, the transcription and the translational machinery, and they use the host, which could be us or the bacteria in this case, then to replicate or produce more little pieces of their nucleotide and also make the proteins that are needed to make more of the little capsids and tails of little, and then it all gets packaged up, and then the cell bursts and spills out a whole bunch more, okay, viruses, and they go out to find other hosts to do their work. And so here you can see an electron micrograph of, here is the bacterial cell wall and membrane, but you can see it's just kind of being obliterated, completely loaded with bacteriophage. Okay. This just kind of demonstrates then what's going on. The assembly, as I said, all these different parts, the base, the tail, the sheath, here's the DNA, capsid, tail fibers. All those little pieces have to be made by the host cell, the bacteria that got infected. Okay? All gets assembled, and then it bursts out. Okay? So the life cycle of the viruses. Are viruses alive? Get some yes, some no. I think it's a really good question. What do you think, Dr. Bowen? Would you say viruses are alive?
Good question.
It is, and there's actually a lot of controversy. I have trouble because some people in the field will say there's more viruses than any other living particle, and I'm like, can't get that out of my mouth, right? But they definitely reproduce. They fulfill some of the requirements that we think of how we define life. So it's tough. They're one of those things that are, I don't know. We'll see. So where do you find phage? They're just about anywhere you find bacteria. Okay? So we've been collecting soil bacteria. We're working specifically on a bacterium called mycobacteriums. So it's a kind of a big group. Okay, anybody know a disease or two caused by mycobacteriums? Besides in my class? Okay, go ahead. Which one? Tuberculosis. You know another one? Leprosy. Okay. So mycobacterium, then some are pathogenic and some aren't. We use a host that's non-pathogenic, Mycobacterium sphegmatis, in the lab. Okay? But what the University of Pittsburgh's trying to do is collect phages that infect Mycobacterium because they're really specific. So some phages infect E. coli, some staph, right? So some Salmonella, some Mycobacterium. Okay? And the purpose of this project is to find phages that can infect and kill Mycobacterium, and how might that be useful to us? Might be a treatment. And more we're seeing antibiotic resistant Mycobacterium tuberculosis strains popping up. Okay? So hopefully this is something we can put in our back pocket. So we're focusing right now on phages that infect Mycobacterium. You find phage hunters everywhere. Okay? This is a group from Florida Gulf Coast, and yes, they spent a lot of time trying to get themselves organized to look like a phage. Okay? But check this out. I mean, TMCC in pretty good company. Okay? This is a pretty important project with lots and lots of participation. So it's pretty cool. So why didn't you know about bacteriophages? They're actually discovered before antibiotics. 10, 15 years before, at least. Okay? Two different researchers kind of discovered them within a couple years of each other. And it was Stalin, Russia, who actually saw potential in this. And so Russia spent the most time researching phages. And today in Georgia, which used to be part of the USSR, right, and now it's kind of independent, but that's where the treatment centers are for phage therapy. Okay. It's centered in Tbilisi. And in Georgia, you have a bacterial infection, you can actually go to the clinic and you don't get antibiotics. You get a vial or an ointment, okay, depending on what you're treating, of phage. You have a choice there, antibiotics or phage treatment. And this is their website, like I said, from Georgia, and people are going all over. Yes, we treat MRSA. Okay. People are coming there from all over the world. We can't get this treatment here. Okay. It's not available yet in our country. So it's pretty cool. So why do we care? We have antibiotics. Okay.
eventually become resistant to all antibiotics. So the only thing theoretically that could kill them for sure in the very end would be a phage.
Yeah. Yes, bacteria are always evolving. And viruses, more lifelike than our chemical antibiotics, if you will, okay, can evolve with them and have for millions, billions of years, right? Exactly. Because we know that we're seeing more and more pathogens showing up resistant to antibiotics. It's pretty scary. And interestingly enough, in 1945, Fleming, who discovered the first antibiotic, notes this: it is not difficult to make microbes resistant to penicillin in the laboratory by exposing them to concentrations not sufficient to kill them. And the same thing has occasionally happened in the body. And by exposing his microbes to non-lethal quantities of the drug, make them resistant. We knew this in the 1940s. And sometimes I think, well, I don't know, what are we thinking, right? So it's not really new. What are we doing in our course? I already mentioned we take soil samples. So we all went out and got soil, bring them back in the lab, grow it up with our Mycobacterium smegmatis, plate them. So this is our capture phase. The next phase then is to start purifying, because if we collect a soil sample, we've got potentially lots of different types of phage, right? But if we're going to really characterize something, we need to narrow that down so there's just one phage, not lots of phages. Okay? And so go into the purification. Once we have a purified vial of our phage, then we do electron microscopy so we can see what it looks like. They don't all look the same. Some have longer tails, some have contractile tails, some don't have tails. Right. So we're going to go to the University of Nevada, Reno, just down the street, and use their electron microscopy facilities to image our phage. We're going to isolate the DNA of the phage, check it through restriction digest, and then sequence it. The phage sequences average about 70,000 base pairs. Okay? So they're not terribly big, okay? Big enough to have some fun with. We're going to get the sequence back. We're then going to do some bioinformatics, annotate, and then publish then our phage at various national databases. Each of the students that isolate a phage get to name their phage, and they're all posted on the phage database. And I should have put that little link to that website, but it's pretty cool. And they're counting. It's amazing. Thousands of phages have been discovered by students since they started this program about 8 years ago. It's just hugely increased what's going on. Right? At TMCC, we're starting this. Our phage hunters are taking Microbiology 251. Okay? So we've kind of messed with the curriculum of the typical microbiology lab. So instead of it all being cookbook, kind of like going in there and you know the instructors knows what's going to happen, and so you know that things are going to work out, we're doing real research. Matter of fact, what's happened all last week, you guys? Failed. Yeah. So we don't know. Tina and I have been like going, woo. It was working so great. And for weeks, I mean, everyone was so successful. And then all of a sudden, we're not infecting for some reason. Okay? It's real research. It's real research. Okay? So what we do, we collect the samples, we put them in a flask with growth media and bacteria, and then we put it in this shaker incubator. This was donated by Sierra Sciences, because we don't really have one. You can see it's kind of old and we have a plywood platform, but it works. Okay? And so we shake them for a day or two. Okay? And you can see Brianna getting hers out, and Shayla and Jocelyn showing theirs off. They're really stinky. Once you get to that point, the whole room smells. It's quite fun, okay? And then what we do is we take them and we plate them on an agar plate, and if we got phage from the environment, then they're going to infect the Mycobacterium sphagmatis and kill them, right? And what it shows up and how we recognize them is you can see the cloudy bacterial lawn, and then the clear spots, those are spots where there was phage that infected the bacteria there and killed it. And so as the bacteria lysed, it cleared a spot in the agar. So these are like our woohoo moments, okay? And some people have worked really hard with multiple. How many samples have you brought in?
I know about 5.
We're in 5. So sometimes you get lucky and the first sample works. Sometimes you just kind of keep bringing stuff in, okay? And Shayla was so bummed after her first try, she wasn't going to try again.
No, but then I brought in another.
Yes, didn't last. It's compelling, you know, you're like, I can do this. Okay, so then we go to purify, and so we do a whole bunch of what are called streaks, you know, so we're trying to thin out the phage now so we can just get one plaque, because in there, theoretically, should be one phage. We go through several rounds of that, trying to get this isolated, okay, and once we do then, we produce what's called a medium titer lysate. So we have a little tube with hopefully a bunch of phage. Really what we need is like milliliters of phage. Okay. All right, but once we get that far and we have our high titer lysate, as I said, we're going to do electron microscopy, isolate the DNA. Some get sent to the University of Pittsburgh where it's archived in the National Phage Database. And here's some examples of students' phages between 2006 and 2009. You said you get to name your phage, so some of them make no sense. Corn dogs, does that not make sense? Okay. So they have different morphologies, different shapes and things. Okay. So we're looking forward to getting to see some of ours. Okay, and as I said then, this is all the wet lab work. After we get the sequence, we go to the in silico. Have you guys heard of in silico? I never heard that term before I saw it here. You know, you have like in vivo, right, which is inside the cell, and then ex vivo or in vitro, outside the cell, and then this coin in silico, which means in the computer, like Silicon Valley. Okay. So in silico. So that's where we're going to start the bioinformatics, where we start taking the language of the DNA and converting that language into the language of proteins, genes, function, right? Trying to figure out how our phage are different or the same as everybody else's. And so there's a lot of stuff on here that's not the point. But what we're trying to do then, here's our DNA sequence, and through the use of computer programs that take different algorithms and things and compare our sequence to other known sequences, we try to figure out what genes these segments might be coding for, what proteins. So some of them are funny. Tape measure is here. That's Tina's favorite. Okay, it has to do with, what do you think?
The tape measure?
Yeah, what do you think the tape measure gene might be about? Yeah, exactly. How long is the tail? How long is the tail going to be in our phage? Okay, so some of the proteins are known, many of them are not known. Okay, so some we're still trying to figure out what they do. What is the point? Okay, if you're interested in this, then we're going to be running our Introduction to Bioinformatics in the spring. It would be open to anybody, not just people that were in microbiology, because at this point, we're going to be just trying to use computer programmings and mathematical formulas and things to try to figure out things about the sequence that we're going to be getting back. Okay? So that's our plug. Okay? And then after we've done this, then the students present the phages at the annual Phages conference in Virginia. So at this annual conference, then students from all over the country get together and show their work. And many of them do talks like I'm doing now. Tina and I went last year. We were like blown away at how good these talks were. The other thing to know about these programs across the country is they're freshmen and sophomores. This program, the purpose of it is to get people doing research and getting involved in science projects early on and not waiting until they're trying to get into graduate school. So it concentrates on freshmen and sophomore, and they do an amazing job. And then the other part is a couple sessions of posters. And it's held at HHMI's Denali Farms Research Park. Tremendous place. It's like, oh my gosh, we should all get to be scientists and live there. It's like amazing. All right. So we wanted to show you some of our work. You said all of our different groups have captured phage. Okay, and what we're working on right now is purifying some of our phage. So some of the students, and Tina, she's ahead of all the rest of us. But of course, she's one of the instructors. So Tina, want to tell us a little bit about the work you've been doing?
Okay, so I was one of the lucky ones. I got phage my very first sample. And it took, very top, right here. So this is for my backyard, my apple tree. And so I named my phage apple tree, and I named it apple tree 2 because over here in the top right, this is a spot plate. So you can see where you have the bacterial lawn, and then you plate different, or spot different phages from what you've collected, and see if it's really actually phage, you're confirming. And so I got all 4 of mine to work, and so I just picked number 2 as mine. So I then started naming it apple tree 2. And so from there, I did a series of streak for isolation. So that's that picture right there. So again, you have your bacterial lawn, and then you streak for isolation like you would a bacterial plate, but you're using virus. And you do this at least 3, I did it 4 times, to make sure that everything on that plate is one virus, one bacteriophage. Because when you're starting out, you're going to have thousands of different kinds. So you want to get somewhere to where you have it pure. And then from there, once I knew that it was a pure isolated phage, I did a series of serial dilutions to get what we call a titer, which is just the concentration of phage in your lysate, which is your tube of bacteriophage. And so as you go more and more dilute, you have less and less phage, and you get to a point where you can actually count them in your spot. And then this just shows, this right here, just shows a picture where there's so much phage, it is completely lysed all the bacteria on the plate.
So you can see the light through it.
Yeah.
On the ceiling.
And then this one right here is just showing a plate, an example of one where you can get plaques. So the clear zones are called plaques. That's where your virus is. And we call them plaque forming units. So you start with one virus, and you get to a point where you have them to use so you can count them. And then that's where you figure out your concentration, your plaque forming units per mil of your solution. That was my apple tree 2.
Thanks, Tina. Brianna, I think you're next.
Yay. Hi, Brianna. I haven't personally found phage yet. It's like the five collections, or 5 soil samples, and still none, but did I get any?
Well, we're still having technical difficulties.
Some work difficulties, but my lab partner got some. They're up here. We got it from a rose bush, or she got it from a rose bush in her backyard. And that was her first time she got it. And then over here is our enrichment plate. So that's our first time we plated it. And so you can see all the what is it?
Plaques.
The lysed bacteria. And so the ones we circled on the plate are the ones we touched and then tried to isolate. So there's our strake 3. And you just try to get the ones to get similar morphology, so you're trying to isolate the only one phage. And that's about it.
That's where you are. Okay, I think Jessica and Kathleen, you guys want to?
I'm Kathleen, and this is my partner Jessica. So I tried to get phage twice and I failed, and then the third time a student brought a soil sample in from Gardnerville.
Mary, who's here.
Oh, she is?
Hiding in the back.
So this is the first time we got phage, and then she didn't get any, so we shared a plate.
Yeah, and so right now we're in the process, how Professor was talking about streaking for isolation. So there's all these phages on there, and we basically, those 2 dots, we pick them, and then we're streaking for isolation. And so you can see here we've streaked twice. And so now we're on the 3rd streak, and that's what we're at right now.
We tried hard. You at least have to do 3 rounds because you really want to just get one phage. If we sequence a mixed population, we're not going to ever be able to make sense of it. So,
Hi everybody, I'm Shayla. I'm Jocelyn. And we just went ahead and called our phage Shirley G because I got the phage from my supervisor at work and it's from her garden, so Shirley G Garden. This is our successful plate from the enrichment that we did. We used the 10 to the minus 3 plate, and
this is our first streak off of the enrichment plate, and as you can see the circle dots, those are our successfully isolated phage that we did the 2nd streak off of.
and see how you can still see some different sizes of plaques there, suggesting that it's still not a pure population. Okay? That we try to get it so that all the plaques will look about the same.
So this is streak number 2. We didn't get as much isolation, but we still got our streak number 3 off of the ones that we circled. We were hoping for the same type of morphology, but it wasn't enough to do the next step.
And then this is where we're at now, streak number 3. We noticed that our macromorphology of the phages got smaller. So we just, and since we have been failing this past couple weeks, we have a lot of them circled. So we've been trying to
we
work
keep
off of
trying.
those. Yeah.
For some reason, our bacteria is resistant to infection. We're not exactly sure why.
So that's pretty much where we are. We're just going to do streak number 4 next time.
Cool. Thanks, you guys. So this map kind of shows across the country where phages have been discovered. And if you notice, it's mostly on the East Coast, right? And none from Nevada. Very few from California, 1 from Oregon, I think. Utah has a big program, so there's quite a few from Utah. But our goal is to get Nevada on the map, right? On the phage map anyways. Okay? And then another opportunity I wanted to share with you is that TMCC recently was gifted, okay, a NASA grant called Community of Practice, where all the community colleges in the state then are getting together to bring undergraduate research to the community colleges. And so out of this grant, TMCC will have 5 scholarships to offer students. It's a $2,000 a semester scholarship over 4 semesters. You don't have to be there all 4, but that's the opportunity. And basically what it is it's a research project, but the real point is learning to think more like a scientist. Doesn't have to be a biologist, right? It's STEM type careers, right? And so it's open. We're working on applications now. They'll be online. And I'm sure the STEM Club will post something once we get things ready to go if any of you are interested. Right now, from our perspective, the plan then is the first semester you would start in microbiology 251 if that was where you wanted, if that's what worked for you. If you weren't on that path, the other entry level then would be in, the bioinformatics class. So if you had an interest in participating in this research, you could either join, and it's only one section of microbiology. We're teaching it Monday, Wednesday, the 12:30. So anyways, you'd have to contact us and ask us what section if you're interested in taking our microbiology section, okay? And then over the summer, this summer we're planning a summer research project, and so we're gonna go to Lake Tahoe and try to capture phage from waters. And we're applying for a Trout Unlimited grant to help support our efforts in this because phage protect fish in the waters from bacterial infections as well. Yeah, you may have heard, like in, I want to say the olden days, but you know, a long time ago, where people would believe that certain waters had healing powers before antibiotics even, right? The Ganges River was really polluted, but people, particularly leprosy actually, was a disease. Of course, nobody knew about microorganisms then, right? They didn't really understand, but what people did understand is bathing in certain waters would help heal. Okay? And we now know that it was phages in those waters that helped heal people's infections. Okay? So we're going to see if we can find some in Lake Tahoe over the summer. The third semester then, if you started with microbiology, would be the bioinformatics course. And then the fourth semester would be an internship in biology where you would continue to work on your research project as well as help other students, mentoring other students, getting them started in undergraduate research. So that's kind of our plan for that perspective. And then this is a video that kind of does a nice job.
Ever since they were discovered some 50 years ago, antibiotics have shielded us from bacterial attack. But as their use increased, so did the virulence and ingenuity of their microbial targets. New strains emerged, bacteria more resistant to antibiotics. Once easily treatable diseases have become hazardous. And now, studies suggest as many as 5 million people die each year from infections that fail to respond to antibiotics. These horrifying microorganisms have even colonized many of our hospitals. Suddenly, we are faced with the very real possibility of a post-antibiotic future, a world of the superbug. Staphylococcus is one of the most dangerous of all bacteria. One by one, it has beaten all of our best antibiotics until only one was left, vancomycin. But last year, Japanese doctors saw the world's first case of vancomycin-resistant staphylococcus. Alarming prospect, but there is one thing that kills even the most resistant bacteria. Unknown to the rest of the world, this magic remedy is cherished in one small country in the heart of the Caucasus Mountains. Phage therapy was pioneered by a maverick Canadian scientist, Felix Durrell, who, although largely self-taught, became renowned for the clarity and elegance of his research. He was an adventurer, who was prepared to go to the ends of the earth to test his theories. But it was in Russia that Durrell got his opportunity to put his theories to work. Stalin, it was said, wanted this magic medicine for his army, and in the Finnish War of the 30s and later in World War II, it was used to protect the Red Army from the dysentery and gangrene that plagued the battlefield. With Stalin's blessing, Durrell founded an institute at Tbilisi, the capital of Georgia, that was dedicated to the study of these magical viruses. Durrell named these healing viruses bacteriophage, which means bacteria eaters, and there is a phage to kill every kind of bacteria. Just as bacteria attack people, these tiny viruses attack them, and they're found wherever bacteria thrive, most often in sewage. Here they are too small to be seen, but the phage are killing off the bacteria, one by one. Phage have extraordinary structures. Their bulk is a head in which their genes are stored. They have 6 legs, or filaments, which attach them to the bacteria, and a tail that works like a hypodermic syringe to infect it with their genes. Inside the bacteria, the phage viruses grow and multiply. Sometimes, as many as 5,000 grow in a single cell. When the new phage burst out, they kill the bacterium, and then each goes on to find another victim. Born in Montreal, where he later returned to both study and work, Durrell was brought up in Paris, where he opened a commercial laboratory and worked at the Pasteur Institute for precise diagnosis. Over the years, the institute trained specialists to find phage, grow them, and turn them into medicine. In the 1970s, when antibiotic-resistant bacteria became a serious problem in Soviet hospitals, people who choose to use phage rather than antibiotics come here to have their infections analyzed and to pick up prescriptions for medicines, tablets, and creams made of phage. They rub the healing viruses into their wounds, drink them for a bad stomach, or swill to cure a gum infection. And every day in the hospital, these viruses are used to save lives. This woman developed gangrene in a wound on her thumb. 5 days ago, the doctors cut away most of the infected tissue. The rest, they treated with phage.
It is ultrasound, and it pushes phage into deeply in tissues.
Phage works wonders in Georgia, and given the chance, it could do the same everywhere. An extraordinary library of phage medicine is held in Tbilisi. It's a national treasure, preserved by the painstaking care of the Institute's scientists. It was built up over the 50 years when problem bacteria were sent here from all across the Soviet Union. Phages were found to fight every new infection, and then they and the bacteria they killed were stored separately for future research. There are phages here to fight hundreds of different infectious diseases. Some are even originals from the 1940s. It's the biggest collection of phage medicine in the world. Phage therapy has been so successful here for so long that it's hard to understand why in the West we have never even heard of it.
So the Russians had no contact with the West, and the West had no contact with them.
Under the Soviet regime, such contacts were officially discouraged, and those whose profile rose too high sometimes had cause for regret. Georgia Aliava, a friend and colleague of Durrell, for instance, was shot during the Stalin era for reasons that are still a matter of speculation. In later years, as the Cold War intensified, the use of phages took on a more strategic note because of their potential use as antidotes to Every time we take an antibiotic or spray one onto a crop, the weakest bacteria are killed. The few strong ones, resistant to the antibiotic, are left to multiply. Resistance is inevitable, and the more antibiotics we use, the worse the problem becomes. This plate of Staphylococcus has been dosed with antibiotics which kill most bacteria. But MRSA, the resistant bacteria, thrive when the sensitive ones die, and they soon take over in a clear example of survival of the fittest. The key problem with antibiotics is that they are unchanging chemicals. Once bacteria develop resistance to them, they become ineffective. But phage is different. The beauty of phage as a medicine is that they are alive, so they can change just as bacteria do. When bacteria evolve resistance, phage evolve alongside. When a superbug appears, a superphage will always evolve to attack it. It's an evolutionary arms race. Phage offers the world a naturally evolving medicine, able to fight off any new bacteria that emerge.
So that's kind of what we're up to in the biology department, and our work on undergraduate research and trying to get more and more opportunities for students to do real life work. All right, so at this point I would say, are there any questions? Yeah.
What kind of bacteria were you killing?
In our lab, we're using Mycobacterium sphegmatis. So it's a strain of Mycobacterium. It's not a pathogen, okay? But the idea being, if we find ones that effectively kill Mycobacterium sphegmatis, they quite likely will kill pathogenic Mycobacterium. That's kind of the hypothesis behind that. Did I answer your question? Okay. Any other questions? Yeah?
So what would happen if a phage evolved to attack human cells then? Because that could also be possible.
So phage probably would never evolve to attack human cells. There's plenty of other viruses that do. But phage could, I mean, there is a potential for it to be harmful in a different way. Okay? So when phage infect, so when the DNA gets released into the bacterium, one of the proteins that nucleic acid codes for is proteins that break up the DNA of the host cell. Okay? And then sometimes accidentally that host DNA can be packaged into the phage. That makes sense, right? Okay? And if by random chance that DNA from the host then starts transferring that to other bacteria, it could be transferring things like antibiotic resistance genes or toxins, so proteins that would be toxic. So in that way, it is possible, so it's not a perfect story, okay? But then again, there would likely be other, phages that could still kill that. So over, what has it been since, it's actually now 50, 60, 70 years of phage research, they haven't seen any evidence of phage being harmful to humans. Mm-hmm.
So like it was stated that viruses and phages are basically in an arms race. Couldn't we technically just end up cultivating phages and just take infections off of basically anyone and just keep injecting, like any bacterial infection, just keep injecting phage into that until we get to the point where we could just fight off basically super strains of every bacteria?
Right. That and potentially that could be.
So is that like the research that's being done?
It's in Georgia, yes. So I didn't show you another video, but there was an American who has a staph infection, so MRSA, okay? And he had a motorcycle accident. And that's how the bacteria got into his leg. And we haven't been able to treat it. And it's gotten so bad, it's been years now, that it's actually started eating away his bone. He's had a couple, 3 knee replacements here because we didn't know what else to do. And it came basically to the point here where his choices were amputation or high dose antibiotics for the rest of his life. And so he actually did go to Georgia and other people from around the world go for treatment too. They have a few different ways of treating. You saw some of them there. That was an old clip. They actually now have some material called Phage Bioderm. So it's like a polymer netting impregnated with phage. And so things like that, they open you up and then like paste the areas with this phage. And then this one, the video shows that they open them all the way up and they put in all this bioderm, but then they have powdered phage, so it's lyophilized with a carrier, and they just sprinkle it all in and then stitch him back up. Okay, and then he does a few weeks of treatment with tubing where they keep pumping, like you're saying, keep pumping phage in. And when he left after a few weeks, it was 10, I think, actually, that he was in the hospital being treated, but there was no more trace of the infection. And so, like they were saying, it can be, you can drink it, it can cure, you know, things in your stomach, tissues. You had one more question?
Yeah, my other question was, even so, due to the fact that phage is only able to target certain bacterial cells, say if you did drink it and it went into your stomach and your body absorbed it, it wouldn't be able to go to the other places, would it? Or is there still a possibility to create phage like that?
That's a good question. I'm sure that we could figure out how to do delivery. That would be like a drug delivery, right? Maybe nanotechnology or something, where we could target, protect the phage from the gut and get them the other way. They are very, very small, okay? 200 nanometers, so they're smaller than ribosomes, okay? So I would say that there would be, I could envision potential for that, okay?
So it seems to me like there's a distinction between bacteriophage and viruses. Are they part of one family? Are they cousins, ancestors?
Good question.
Do we know?
Yes. Okay, so viruses are one big family. Okay? A virus is defined by something that has nothing more than a piece of nucleic acid, either an RNA or DNA, and then a protein coat, okay? And then some other appendages, right? We saw what bacteriophage looked like. If we think about viruses that infect bacteria, bacteria have a fairly rigid cell wall in addition to a cell membrane. So they have to have some machinery that can drill through that thick wall to actually get inside, okay? Viruses that affect us are animal cells where we just have the plasma membrane. They don't need that much, okay? So animal viruses could be RNA or DNA, right? Single-stranded or double-stranded, lots of versions. Mostly they all are covered with a protein. Often they're enveloped by membrane, okay? And that's kind of how they elude the immune system, but it's also how they can enter. So animal viruses, similar, of course, but different delivery mechanisms. They don't have to penetrate. Instead, they'll often just kind of merge with the membrane of the host and deliver their nucleic acid.
So an animal virus is maybe more simple than a bacteriophage?
Yeah, more simple in some ways and maybe more complicated in others.
We just don't have as much as a defense for
right.
Like bacteria does.
Right. But animal viruses then, and these two, they all are really involved with specific receptor-mediated attachment, which is why one phage will infect Mycobacterium and not E. coli. Another will infect Salmonella and not Staph. So that's kind of why there's this whole big bank of phages, and so they need to find out what kind of infection you have because the treatment becomes very specific. Another cool thing about that, though, is think about how our antibiotics work. For the most part, they kill off a lot of different, most of them are broad spectrum, right? So they kill off our good bacteria as well and compromise our immune system. With phage treatment, you can kind of narrow that down so you're only then killing off at least all the Mycobacterium, right? And not the good bacteria that might be in your system. Yeah. Any other questions? Yeah.
So bacteria would develop first and phage develop after that?
Chicken and the egg.
The chicken and the egg, yes. I know. I don't think that's something that anybody knows. So the theory would probably be that viruses are perhaps fragments of prokaryotic organisms, you know, that somehow evolved together. So, but then again, maybe the virus is formed in the, I don't think we have a clear understanding of that. Yeah.
If you've already taken 251 this summer, is that when that class is offered for the Tahoe
Yes.
research? Okay.
Yes, yeah. So, yes, so, and we have the scholarship as one idea, but if you're interested in joining us for summer research, you don't have to have a scholarship to do that. Okay? Yeah, we'll you know, send people over, and Dr. Bowen left, right? But he's volunteered, he has a boat. So he's going to take us around Lake Tahoe, because we got to get samples. I like Emerald Bay, that's like my favorite place. And you can't like get there very easily unless you have a boat. So anyways, yeah, absolutely. So if you're interested in undergraduate research, then, you know, get in touch with me, because we're trying all different kinds of things. Okay, any other questions? Okay, well, thank you. And thank you guys for coming.